Welcome to the Indian Biological Sciences and Research Institute's workshop on in-silico drug designing, from target identification to lead optimization using molecular docking. This session is presented by Ms. Sheetal Mahato, PG Bioinformatics Research Associate at IBRI. Over the next 1.5 hours, we will cover the full workflow, including a hands-on molecular docking practical using Molegro Virtual Docker, or MVD. Here is our session roadmap. We have 13 building blocks today, starting with the basics of drug discovery, moving through in-silico drug design, protein and ligand database retrieval, molecular file formats, protein and ligand preparation, active site identification, and molecular docking concepts. In the second half, we will run a live docking demonstration in Molegro Virtual Docker, interpret the results, understand binding affinity, predict ADMET properties, visualize protein-ligand interactions, and finish with real-world applications. Let's start with the basics of drug discovery. Drug discovery is the multidisciplinary process of identifying a chemical entity that can safely and effectively modulate a biological target to treat disease. The pipeline moves from target discovery to hit identification, lead optimization, preclinical studies, clinical trials, and finally regulatory approval. This typically takes 10 to 15 years, costs between $1 and $2.6 billion, and around 90% of candidates still fail during trials. This is exactly why we go in silico. Computational pre-screening filters out weak candidates before costly wet lab and animal testing, shrinks the hit-to-lead timeline, lowers R&D expenditure, and enables rational, structure-guided design instead of trial and error. Now, an introduction to in-silico drug design. In-silico simply means performed via computer simulation. Computer-aided drug design, or CADD, uses computational models to predict how molecules behave and bind. Core techniques include molecular docking, virtual screening, QSAR, pharmacophore modeling, homology modeling, and molecular dynamics simulation. There are two broad approaches. Structure-based design, which uses the 3D structure of the target protein from X-ray, NMR, or AlphaFold, fitting the ligand directly into the binding pocket, and ligand-based design, which uses known active and inactive ligands when the target structure is unavailable, building pharmacophore or QSAR models instead. The advantage of going in silico is faster iteration, lower cost, fewer animals used, and the ability to virtually screen millions of compounds before a single wet lab assay is even run. Next, protein and ligand database retrieval. For protein structures, we rely on the RCSB protein DataBank for experimentally solved structures, UniProt for protein sequences and functional information, and the AlphaFold database for AI-predicted structures when no experimental structure exists. For ligands and compounds, we use PubChem, DrugBank, ChEMBL, and ZINC for ready-to-dock compound libraries. When choosing a good protein structure, look for a resolution better than 2.5 angstroms, the presence of a co-crystal ligand confirming a real binding site, structural completeness with no missing residues or loops, and a low R-free value, which indicates a more reliable model. Molecular file formats matter because the same molecule can be saved in different file types, each storing slightly different information. For small molecules, or ligands, we commonly use SMILES, which writes an entire molecule as a single line of text, the .sdf format, which adds 3D coordinates and chemical property data, and .mol2, which adds atom types and partial charges needed for docking. For proteins, the classic format is .pdb, containing 3D atomic coordinates, and its modern, more detailed successor, .cif or mmcif. Here is the anatomy of a PDB file. Its columns encode the atom index, the residue, the chain, and the 3D coordinates for every atom in the structure. And here is a raw SDF record, showing the atom coordinates and bond table for a single ligand. Raw structures downloaded from databases are never docking ready. Each one needs careful cleanup first. For protein preparation, we remove crystallographic water molecules and unwanted heteroatoms while retaining essential cofactors and metal ions, Fix missing residues, atoms, and loops. Assign correct bond orders and formal charges. Add polar hydrogen atoms often missing in X-ray structures. Assign optimal protonation states at physiological pH. And energy minimize the structure to relieve steric clashes. For ligand preparation, we convert the 2D structure to a realistic 3D conformation. Add explicit hydrogens. Assign the correct protonation state and tautomer. Assign partial charges, such as gas tiger charges. Energy minimize to a stable geometry, and convert to a dockable format like PDB-QT. Common tools for this include OpenBabel, Avogadro, Autodock tools, UXF-Chimera, and PyMOL. Locating the binding pocket where the ligand is expected to bind is critical. An inaccurate site produces meaningless docking results. There are three main approaches. Using a reference ligand, where a co-crystallized ligand's position in the PDB structure marks the known site. Cavity detection, where algorithms like the MVD detector, CASP, F-pocket, and sitemap scan the protein surface for pockets. And conservation analysis, where residues conserved across homologous proteins often mark functional or catalytic sites. This brings us to the grid box. The search volume that MVD or Autodock explores, defined by a center coordinate and a size, set around the detected cavity or reference ligand position. Targeted docking searches a known pocket and is faster and more precise, while blind docking searches the whole protein when the pocket is unknown. Now, molecular docking concepts. Docking predicts the preferred orientation, or pose, of a ligand inside a protein's binding site, and estimates the strength of that interaction. Three components drive this. The search algorithm, which explores possible ligand poses using methods like genetic algorithms, Monte Carlo, or incremental construction. The scoring function, which ranks the generated poses by predicted binding energy using force field, empirical, or knowledge-based methods. And the binding model, which can be lock and key, a rigid fixed shape fit, or induced fit, where the pocket adapts to the ligand. Docking types range from rigid, where both protein and ligand are held fixed, to flexible ligand, the most common approach, where ligand torsions rotate freely, to flexible protein ligand, where pocket side chains also move. The output of a docking run is a ranked set of predicted 3D binding poses, each with a score, along with interaction details for downstream analysis. This diagram illustrates the lock and key versus induced fit models, the classical concepts behind docking's binding assumptions. Now let's introduce our hands-on tool, Molegro Virtual Docker, or MVD, an integrated software platform for predicting protein-ligand interactions. It is built around the proprietary MALDOC algorithm, a search engine combining an evolutionary genetic algorithm with guided differential evolution. MVD provides an end-to-end workflow in one interface: structure import, preparation, cavity detection, docking, scoring, and pose visualization. Key features include automatic cavity prediction, the MALDOC scoring engine, a built-in 3D viewer, and a beginner-friendly GUI requiring no scripting for a full run. We use it for teaching because a single guided pipeline takes learners from a raw PDB file to a scored, ranked, and visualized docking result. Let's set up the docking environment in MVD in 4 steps. First, import the protein. Use file, import molecule, and either fetch directly by PDB ID or load a local.pdb or .cif file. Second, prepare the structure using the preparation wizard. Remove waters and unwanted heteroatoms, assign bonds and charges, add hydrogens, and keep essential cofactors. Third, detect cavities. Run MVD's grid-based detect cavities algorithm, which automatically highlights candidate binding pockets on the surface. Fourth, define the search space by selecting the chosen cavity, or building a template sphere around known active site residues, to set the docking search volume. Now let's run the actual docking job, in 4 more steps. Fifth, import and prepare the ligand. Load it in .sdf or .mol2 format, add hydrogens, assign charges, and mark rotatable bonds as flexible torsions. Sixth, configure docking parameters. Set the algorithm to MALDOC simplex evolution, Choose the number of runs, for example 10, along with maximum iterations, population size, and energy grid resolution. 7. Run the docking search. MVD's guided differential evolution explores poses within the cavity, scoring each with the maldoc scoring function. 8. Export and save results. Poses are automatically ranked by score. Save the session as an MVDB file, and export the top poses in SDF or PDB format for downstream analysis. Now, interpreting docking results. MVD ranks every generated pose, and reading the score table and interaction map correctly is what turns a docking run into a real conclusion. The maldoc score measures the total estimated interaction energy of the pose. A more negative score means stronger predicted binding. The re-rank score is a refined rescoring of the top poses, used to distinguish close maldoc scores. RMSD, in angstroms, measures the deviation of the docked pose versus the co-crystal ligand. Under 2 angstroms is considered a successful redocking. Ligand efficiency is the binding score normalized by heavy atom count. Higher efficiency suggests a more optimizable lead. Beyond the numbers, always inspect visually for hydrogen bonds, hydrophobic contacts, pi-pi stacking, electrostatic interactions, and steric clashes. Let's talk about binding affinity and scoring functions. Binding affinity measures how strongly a ligand binds its target, and docking reports it as a score, usually in kilocalories per mole. A more negative score means stronger predicted binding. For example, a docking score of -9.5 kilocalories per mole indicates tighter binding than -6.0. Related measurements include delta G, the binding free energy, KD and Chi, the dissociation and inhibition constants, where lower means tighter binding, and IC50, the concentration needed for 50% inhibition, where lower means more potent. Scoring functions evaluate hydrogen bonds, hydrophobic contacts, electrostatic interactions, and van der Waals forces. Remember, these scores are estimates best used to rank candidates. Always validate top hits with laboratory experiments. Strong binder is worthless as a drug if it can't reach the target or turns out to be toxic. This is where ADMET prediction comes in, screening for real-world viability early. ADMET stands for absorption, distribution, metabolism, excretion, and toxicity. Absorption is assessed using Lipinski's rule of 5 and CACO2 permeability. Distribution considers blood-brain barrier penetration and plasma protein binding. Metabolism looks at interaction with CYP450 enzymes and metabolic stability. Excretion covers clearance rate and elimination half-life. And toxicity evaluates hepatotoxicity, mutagenicity, cardiotoxicity, and LD50. Common prediction tools include Swiss ADMET, ADMET SAR, PKCSM, and Protox Roman 2. ADMET filtering is applied after docking to shortlist hits that are not just potent, but also drug-like and safe. Turning numeric scores into a visual story is essential for validating and communicating the binding hypothesis. This is protein-ligand visualization. Popular tools include PyMOL for high-quality 3D rendering and publication figures, Discovery Studio Visualizer for 2D and 3D interaction diagrams, UXF Chimera and Chimera X, and MVD Zone built-in 3D viewer for inspecting poses immediately after docking. What should you look at? The 3D binding pose, showing the ligand's orientation inside the pocket. The 2D interaction map, showing hydrogen bonds, hydrophobic patches, and distances, and the surface or cavity view, showing the shape and electrostatics of the pocket. Visual inspection confirms the pose makes chemical sense, reveals which residues drive affinity, and guides structure-activity relationship-based lead optimization in later design cycles. Finally, let's look at applications of these methods in modern drug discovery. Virtual screening ranks millions of library compounds computationally before any lab assay. Lead optimization uses structure-guided modification for higher potency and selectivity. Drug repurposing tests approved drugs against new targets, as we saw during COVID-19 screening efforts. Target validation confirms a protein is druggable before investing in a full program. vaccine and peptide design models, antigen receptor and peptide target interactions. And overall, these approaches cut R&D spend and minimize costly in vivo experimentation. To summarize, here is the complete In-Silico pipeline, starting from target and database identification, through structure preparation, active site detection, MVD docking, results and ADMET evaluation, and finally visualization and application. In-Silico methods de-risk and accelerate early drug discovery before any wet lab work begins. Careful protein and ligand preparation, along with accurate active site detection, directly determine docking quality. MALDOC scores, re-rank scores, and interaction maps together, not any single number, tell the real binding story. And ADMET filtering ensures a computationally good binder is also a viable, safe drug candidate. Thank you for joining this workshop. We now open the floor for questions and discussion. This session was brought to you by the Indian Biological Sciences and Research Institute. You can reach us at +91 99995 09892 or 99714 41910 by email at info@ibri.org.in or online at www.ibri.org.in. Once again, thank you to our speaker, Ms. Sheetal Mahato, PG Bioinformatics Research Associate at IBRI. Little about IBRI. The Indian Biological Sciences and Research Institute, based in Noida, is an ISO 9001:2015 certified institute training students and professionals across India and over 50 countries in bioinformatics, computational biology, and drug discovery. We offer a 3-month certificate program, a 6-month certificate program, and a 12-month comprehensive program. IBRI also offers project training and free dissertation support in bioinformatics and biotechnology, corporate training programs for life sciences and industry, thesis writing and medical scientific documentation guidance, and both classroom and distance learning tracks, along with hands-on workshops in silico drug designing, next-generation sequencing, molecular modeling, and AI and machine learning. Before we close, here are our upcoming workshops at IBRI. Registration is open now, and seats are limited. Visit ibri.org.in/workshop.html. On the 23rd of August, 2026, join our session on drug regulatory requirements of rest of world nations, covering regulatory frameworks and submission requirements across global pharma and biotech markets. On the 30th of August, 2026, join our Introduction to AI and Machine Learning workshop, a hands-on coding session covering ML fundamentals and real-world applications in life sciences and drug discovery. For contact and registration, call +91 99714 41910, email info@ibri.org.in, or visit www.ibri.org.in. Thank you for watching.